So I'll continue with this, this idea that we've been talking about in risk of specifically carbon nanotubes, and a little bit about perception, mostly about toxicology, and personal experiences that I've gotten into in terms of being a scientist, trying to be as straightforward as I can about reporting what is accurate in terms of science. Before I came to NC State about a year ago, I worked for the federal government, the NIH, and it's a bit of an exaggeration, but as toxicologists there, we're led to believe that everything's toxic. And then I worked for an industry-funded institute for about the next 3.5 years, and there we're led to believe that everything is not toxic. So I feel like I'm landing in the middle here somewhere. When I was at this industry-funded institute, that I was given the opportunity to begin working on some nanomaterials. I'm a lung biologist. I work on pulmonary fibrosis. I actually started working on asbestos about 20 years ago, so I have some perspective on that. Oops. Let me go forward. I'm hitting all sorts of buttons I'm not supposed to here. Ah, thank you. Ah, left click. Thank you. So, in general, as we all know already, nanomaterials offer benefits and risk. The benefits are potentially numerous. Structural materials could be the solution to a lot of our energy problems. Bioremediation, potentially very important contributions to medicine, imaging, drug delivery. The benefits go on and on. So I don't want to underemphasize that. Again, as toxicologists, we're trained to look at risk, but in this context, we also must look at the benefits. So the risk, as Tara has brought up very nicely, involve potentially environmental contamination and toxicity to ecosystems. We don't really know, I was going to ask you this question, Tara, but we don't really know what the concentrations are that organisms in the ecosystems are going to be exposed to. We don't really know what the concentrations that individuals in occupational settings will be exposed to. And we don't really know what consumers will be exposed to a variety of different nanomaterials. So there are clearly unanticipated effects on human health that we're just beginning to learn about. So the potential issues when we talk about nanomaterials and health sciences are human exposures to nanomaterials. Their unique properties that make them very useful for a wide variety of different applications, electronics, structural materials, and so on, also make them potentially hazardous. That's going to alter their translocation once they're taken up by an individual, whether it be a human or another organism. And this is going to alter the potential risk for disease. Again, nanomaterials in the environment, what happens when they get in the air, the water, and the soil, and we don't know. Nanotechnology in medicine, as I already mentioned. Quantum dots for imaging are bound to be very useful. They could have potential toxic side effects. Drug delivery, carbon nanotubes, and a variety of other fullerenes and other nanomaterials are already being investigated very heavily, and a lot of money being put into this for drug delivery, for cancer. If you Google, for example, carbon nanotubes and cancer, you'll get this study that we've been talking about that I'll mention in a minute. But you also get some very cutting-edge type work where carbon nanotubes have been used to burn out cancer cells, and they can be heated up. And if they're in the right place, they'll kill a cancer cell. So there could be very beneficial effects there. Also for tissue repair and tissue remodeling, growing cells for transplants and bone remodeling and all sorts of things that could be very beneficial. So, as I mentioned, I began working on particles and fibers a long time ago when I started working at the NIH under Arnold Brody, who worked on asbestos fibers, still works on asbestos fibers here at NC State. One of the things that I teach in one of my toxicology lectures on lung toxicity is that whether they're fibers or whether they're particles, there are some basic rules that dictate toxicity. For fibers, increasing fiber length is directly correlated with increasing toxicity in the lung and is correlated with decreasing clearance. So the problem with asbestos fibers are the long ones. Those are the ones that stay in the lung. If they're longer than 10 microns or 12 microns here, which is about the width of this cell I've drawn here. This is a lung macrophage, which I'll tell you more about. If it's longer than the cell is wide, the cell can't handle it. It gets frustrated and it tries to clear it, and it can't. The fiber stays in the lung for years. 20 years later, an individual ends up with asbestosis, or which is a fibrotic disease, or mesothelioma, which is a cancer. The two are different. With particles, decreasing particle diameter is directly correlated with increasing ROS, or reactive oxygen species, the generation of oxidants, and increasing toxicity per unit mass. So the idea and one of the worries initially with small particles, and this started with air pollutant particles, the work by Günter Oberdorster and others, where ultrafines, were initially called ultrafines, they're now nano-sized air pollutant particles, are thought to be more toxic because of their increasing surface area. So this is an issue. And then things become more complicated when we talk about engineered nanomaterials. So for asbestos, this is a scanning electron micrograph that was taken about 20 years ago by Dr. Arnold Brody. These are macrophages here in the lung. They're on the surface of the lung epithelium. This bar here shows you about 10 microns, so we're not in the nano range now. This is an asbestos fiber. It's very long. It's probably about 100 to 200 nanometers wide, I'm guessing, just by the length of it here. And there are 3 macrophages that are trying to share it here and trying to clear it from the lung, and they can't. So it's clearly known that these fibers cause lung disease. And then there are the benign types of particles. These are carbonyl iron spheres. This is a macrophage in culture that's taken up these 1 micron or so, 1,000 nanometer wide iron spheres, and it's packed full of them, but they're not that toxic. The macrophages can clear them out of the lung pretty quickly. So when we get down to this very small size range, this little dot here, this is 25 nanometers, if you can see that dot. And then if we look at something that's built on that scale as a fiber, this would be, say, a multi-walled carbon nanotube. So it has a high aspect ratio, meaning it's very long and not very wide. It's like a fiber. So they're on this spectrum for assessing lung disease and whether nanomaterials or anything else can cause cancer or fibrosis in the lungs. We look at factors or environmental agents that are either no or low risk. These are things like carbon black, toner cartridges. How often do you worry if you're changing a toner cartridge on your printer, you get a little dust on your hands? Do you really worry about that? Are people worried about that? No, not really, because it's fairly innocuous. Mount St. Helens ash from the volcano, if you remember the volcano that exploded, unless you were directly downwind or down the explosion site, you would have been killed. The dust itself is very non-toxic. And then on the other end of the scale, there are very defined risks. The pandemic of asbestos fibers, over the last 50 or 60 years, people were exposed to a variety of different types of asbestos.